Mouse lemurs make up the genus Microcebus. They're small matrilocal primates, and they have low paternal care, and they're highly speciose. You can see on the right, there's a large map here depicting where they're found across the island of Madagascar. And they're also likely sperm competitors. We know this from mostly behavioral observations. They have multiple male populations. The females are, like I said, matrilocals, but the males leave, and kind of females control the, like many primates, females control the nest, or the home. And then they also have morphologically large testes in season. So my main question is, are these two things related? And broadly, very, very broadly, have rapidly evolving sperm-related genes contributed to the high species diversity found in mouse lemurs, or the genus Microcebus. But more specifically, the part of this I want to tackle first was a more narrow hypothesis of sperm competition and the accompanying rapid evolution of sperm genes, and has it left a signature of non-synonymous amino acid change across genomes within this genus. So for data, we now have a Microcebus moranis genome. It's a chromosome-level assembly, and it is annotated last year. That's important because now we can begin to ask functional questions. And so, along with this one species down here, we sequenced several other species at low coverage with an Illumina HiSeq. For methods, we used Bowtie2 to map sequence data from birthday, which is one of the other species in the genus, to moranis. SAM tools to call a consensus sequence for those birthday reads, determine the SNPs between the species after an alignment with Frank, and then compared from 150 sperm genes from the moranis genome to randomly selected genes throughout the moranis genome. Most of the comparison was done on Python. I'd like to go ahead and thank Matt Dubin, who is a rotation student in our lab, who helped a lot with the Python end of the pipeline. So the results. So what the basic question was to compare the NDS for the sperm genes and the non-sperm genes. And in the presence of sperm competition, we would expect that the sperm genes to have higher non-synonymous changes than the randomly selected genes. And that's what we indeed did see. So the sperm genes here colored in blue are to the right. They have a higher log DNDS. It's a significant difference. And the random genes are in red. Another way to look at the same plot is to plot it in 2 dimensions, where you have the count of non-synonymous SNPs on the y-axis, the count of synonymous SNPs on the x, and anything falling in the top part of the diagram has more non-synonymous changes than synonymous. And if you plot a line to each set of data, the higher the slope of the line, the more non-synonymous changes you're seeing. So you can see the blue line does have a higher slope. So that actually doesn't really approach function, because the way I was defining a sperm gene was a gene that literally said sperm anywhere in the definition. So that's how we got to 150. However, there's been some other work done in primate sperm genes, and I was able to find 10 genes that were analogous to other primate work, 3 of which were sperm construction, so things that were involved in, I guarantee the function was involved in building the sperm. And another 7 that was sperm egg fertilization. And in the presence of reproductive isolation, we would really expect that these fertilization genes would be the ones that would be changing. If these species are in allopatry and they have a non-synonymous change that's going to prevent, or create new species and prevent hybridization, it's going to be in those genes. So I plotted those back onto this, and as you can see, there's more non-synonymous changes in the green dots here, the fertilization genes, than the construction genes. You can kind of see the underlying plot here. The gray line is a 1-to-1, kind of at the NDS equals 1 for reference. Obviously, this is still a little bit early in the work, and I'm hoping to characterize the exact function of more genes, but in summary, Microcebus overall sperm genes have more non-synonymous changes than non-sperm genes with this plot, and then there are more non-synonymous changes in genes involving sperm egg fertilization as well. And going back to the bigger question, it seems that changes in sperm genes have potentially contributed to the high species diversity among moranis, or among Microcebus. So this is something that, like I said, does need further work, but this is what I would expect to see if these 2 things are indeed related. I'd like to thank my advisor, Dan Yoder. Like I said, Matt Dubin, who did a rotation in the lab, where we did the sequencing. Peter Larson, who's the postdoc in our lab, the rest of the lab, NSF, my committee, the Lemur Center, do better. Thanks.